Okay, this is Bio 181, Chapter 5 on membranes and transport. And now we're going to be talking about active transport. So active transport, think ATP. ATP is our energy molecule of the cell. So in active transport, it's going to require energy. And so it requires energy to move things from an area of low concentration to high concentration, right? So we're going from low to high. So it's the opposite of passive transport. So if you have, say, a whole bunch of stuff in the cell and you want to get more stuff in there, just random motion bumping around, it's not going to just go in. So you're actually going to have to add energy to get it in. So you're going to require energy and you're going to go from low to high. Okay. All right. So that's what's shown in this image here. You've got this molecule ATP. Again, here, this is our cell membrane. It's our phospholipid bilayer. You can see here's the phospholipid 1. There's another phospholipid. Okay. We can see that we also have some protein transporters here. So here's one. This is a protein. Okay. And it's transporting a couple different molecules, one in one direction, one in another. So that would be an antiporter. Okay. And it's going to be transporting them against the concentration gradient. So if we were to count up the molecules, yeah, okay, so there. It's not doing a great job here. It's not a huge difference, but basically, if I was to emphasize this, we would be adding a whole bunch of molecules, green molecules, from an area of low down here to high up here. That would require ATP to go in that direction. Okay. And then similarly, if we had a whole bunch of molecules in here, we had a whole bunch of purple molecules, so ions in here, we had a whole bunch, right? And we were going to take those molecules and move them in this direction from low to high. Then that would also take energy. Okay. Okay, so active transport, it's going to be used anytime that we have an ion or a molecule that's being transported through a membrane, okay? And it's going to go against the concentration gradient, against the concentration gradient. So again, we're moving from an area, say out here, if it's high, and in here it's low. If you're trying to get it out to where it's higher, this is going to be active transport, right? It takes energy to go from low to high. So just emphasizing that again, okay? Similarly, if there happens to be an electrochemical gradient, then it's also going to take energy. So say you're going to go from an area and you're moving something that's positive into an area that's even more positive. Well, you know, if it's the same charge, those are going to repel. So if you're trying to move something into an area of the same voltage, that's also going to take energy, okay? Into that same, either more positive if it's positive or more negative if it's negative. Okay, so ATP is used. Next. Okay. So what I wanted to show you here, this diagram is looking at active transport And so it's similar to the other one, but I like the fact that we've got multiple proteins that are shown in the plasma membrane, right? And it shows that process of using the ATP here, getting the energy out and transporting it. It also shows the gradient over here. So here's the sodium gradient. It's more sodium molecules are on the outside and there's fewer sodium molecules on the inside. So if we're moving them from the area where there's less to the area where there's more, that's going to take ATP. And similarly with this potassium ion, if we're moving it from an area where there's less to an area where it's more, which is shown over here on the right, that's also going to require ATP. Okay. And this example is a sodium potassium pump. Okay. So sodium, potassium, they're both positive ions. They're being moved in opposite directions. So it's an antiporter. Every time it goes through the cycle, it's moving 3 sodium ions out and 2 potassium ions in. So again, this is an antiporter, and it's very important for establishing electrochemical gradients, which are going to be important for sending nervous signals in the body. And this here really is just the same thing. So this is in our book. It's a different diagram, but I wanted to orient you and just tell you that it's the same thing. Here come the sodium ions, right? The ATP is utilized. They're dumped on the outside. Then we get the in this case, we're getting the potassiums coming in, and then they are going to be moved to the inside. Okay, last concept for active transport is bulk transport. So bulk transport, we're going to be bringing large items, okay? So they could be really big molecules okay, just a lot of them, many molecules, right? Many molecules. Okay, so if we want to bring big stuff or a whole bunch of stuff in or out of the cell, that's called bulk transport. And if it's going in, endo is inside. So this is bringing in. Cyto is the cell. So this is bringing into the cell. Exo is going out. Okay. And again, of the cell. Okay. So shown here, exocytosis. So you've got down here, you've got some proteins that have been made. And here's a vesicle. Maybe they were made in the rough endoplasmic reticulum, and then they were modified in the Golgi apparatus, and then they're now going to be secreted. So it's moving in a vesicle, a membrane-bound compartment, and that compartment is moving toward the outside of the cell. Then it's going to merge with the outside of the cell, and the contents inside are going to be dumped outside. What I really like about this is you can see that it's the same material that is actually surrounding the vesicle that's going to then merge with the plasma membrane. So all of this stuff is a phospholipid bilayer, right? This is all a phospholipid bilayer. Lipid bilayer, okay? And if you remember, the vesicles are part of what we call the endomembrane system, membrane system. So the membrane on the outside, this is the cell membrane out here all along the outside, that's membrane. And then endo, again, is inside. So it's pretty cool. It's just merging that same material is then going to just merge with the outside of the cell. And cells can actually grow larger this way by creating plasma membrane material, phospholipid bilayers, being extended by just merging with that plasma membrane. Okay, opposite of that, here you have a huge thing. Maybe it's a big, large piece of food, or maybe this is a bacteria and it's engulfing another organism, even an entire organism. It can surround it. This is called an endocytic vesicle. So it's going to surround it inside. and then it's going to bring it in. And if this is going to be digested, so maybe it goes to, you guys remember the name of the component of the cell that has digestive enzymes? Lysosome. Okay, so it's going to merge maybe with the lysosome with digestive enzymes, and they're going to become one, and then this is going to be degraded. Okay. All right, so that is bulk transport. Does require energy. Okay. Inside is endocytosis and outside is exocytosis. The one last concept here is that sometimes for endocytosis, it requires receptors on the surface of the membrane to be signaled. And that signal, when something binds to it, is going to initiate the endocytosis. So there might just be these receptors, these proteins that are on the outside of the cell. They're just sitting there waiting. And if all of a sudden they start receiving signals, then that's going to tell the cell, hey, let's surround all of this and bring it into the cell. And again, exocytosis, again, here is our vesicle, and then it's merging with the extracellular fluid. All right, that's it for this video.